All right. We talked about two kinds of photoinhibition. So when I say photoinhibition, I mean when the leaf gets too much light. Too much light can damage a leaf. You can have dynamic photoinhibition, which is temporary. It's not really damaging, but it does have some effects on photosynthesis for a leaf. It doesn't have an effect on the maximum photosynthesis, and so the leaf is still able to perform photosynthesis at maximal rates in high light, even with dynamic photoinhibition. But there is a shift in the compensation point, meaning that the relationship between photosynthesis and respiration shifts so that there is more respiration per unit photosynthesis, and also the respiration rate becomes higher or more negative. So the respiration rate becomes a bigger source of CO2 to the atmosphere, or from the leaf, or to the atmosphere. Chronic photoinhibition is when the leaf has been suffering through so much excessive light load that the photosynthetic apparatus has been damaged. And you can tell that the photosynthetic apparatus has been damaged because the maximal photosynthetic rate in saturating light or in full sunlight is much lower than it would be if it were under optimal light conditions or even under dynamic photoinhibition. There's also the change in the compensation point for both kinds, as well as a change in the dark respiration, an increase in dark respiration for both types of photoinhibition. The key concepts from this lecture, or from this part of the lecture, which we've already gone over, we have maximum photosynthesis, compensation point, where photosynthesis and respiration compensate each other, and dark respiration. Quantum yield, which we talked about a lot during the first half of this subject, is the slope of this curve at low light. So it's basically the ability of that plant to take up light when there's not a lot of it. So this is when the plants are light limited, that's when the plants are CO2 limited. We talked about leaf thickness, remembering that leaf mass per area is just the inverse of specific leaf area. They both describe how thick the leaf is. Now we're heading into some of the lectures that were quite a lot more in depth. So starting with stomata, and we start with the guard cells. Stomata are in the epidermis of the leaf, made of two cells called guard cells. They have chloroplasts because, as I'm about to show you, they actually use light energy through photosynthetic processes to generate the turgor that will cause the cells to open, or cause this aperture to open. They lack plasmodesmata because they need to generate pressure within the cell, and so you can't have any way for the water to escape out of the cell. So they lack plasmodesmata. They can be kidney-shaped like you see in dicots, or they can be dumbbell-shaped like you see in monocots or grasses. And what we really want to focus on here are these cellulose microfibrils. These cellulose microfibrils are arranged in a way so that as the turgor pressure in the cell starts to increase, the cell deforms its shape, and that's what creates this opening or pore in between the guard cells. So the example I used during the lecture was, imagine you have one of those long tubular balloons, and you wrap some tape around it like these microfibrils at regular intervals. When you inflate that balloon with that tape around the outside, it will actually bend into a curve very much like the shape of one of these guard cells. And so that's how it does it. At the cellular level, what happens when the stomata start to open? So these proton pumping ATPases are activated. The way that they're activated is the photosynthesis carried out by the chloroplasts, say we're particularly talking about in the morning. The chloroplasts perform photosynthesis, which generates malate and ATP. That ATP is then used to pump protons out of the guard cell. When you have a large enough proton motive force between the outside of the guard cell and the cytosol, that turns on these potassium pumps, or these potassium channels. And then you get potassium moving into the guard cell. As potassium moves into the guard cell, the turgor goes up because the solute potential goes down. So your solute concentration goes up, solute potential goes down, builds up turgor inside that cell and causes it to deform. And the way that it does this is by coupling these proton pumping ATPases to these potassium channels.